Today we'll start with the sample problems for week 6 of NPTEL Experimental Biotechnology. The first question for today's class is: dash is used to elute the analyte during affinity chromatography. Option A counter ligand, option B ligand, option C receptor, option D counter receptor. You can think about this question for 2 minutes. To start with, you can tell us what affinity chromatography is. We'll take it from there. Okay, so affinity chromatography is this technique where we try to separate our molecules of interest based on chemical affinity. So by chemical affinity, what I mean is specificity. Like antibodies and antigens have specificity, there would be some chemical species that have affinity for something, and we would incorporate that region of having affinity in our molecule of interest. A simple example that we all are aware of is histidine tag, hexahistidine tag, or octahistidine tag, where 6 to 8 residues of histidine amino acid are covalently linked, and in the 5 prime of your DNA, which is N terminal of your protein, or in the C terminal of your protein. Now what this does is it forms a 3-dimensional geometry where it can either interact with, it can create coordination with nickel and cobalt as well. So this is one example of affinity chromatography. Now the thing with affinity chromatography is, first we have a support or the matrix where we have something embedded. Like in nickel NTA chromatography, we have these nickel containing resins. Now our proteins containing histidine tag will bind to it. Now the question is, how do we elute this? So to elute this, we need something that binds even stronger than our protein of interest. In that case, we use something called as imidazole, which uh, mimics the histidine rings. So higher concentrations of imidazole would compete with our protein for the nickel. Hence, our protein will be eluted. This is how affinity chromatography works. So in this case, imidazole is rather a counter ligand. It competes with our protein to be able to bind to the nickel NTA beads. So the right answer for this question is option A, counter ligand. This is just a description of how affinity chromatography works. The next question is, antibody antigen receptor ligand pairs are used in dash, and four different options are given here: bioaffinity, gel filtration, ion exchange, and pseudo affinity. You can think about this question for a couple of minutes and then we'll discuss this. Okay, like we discussed in the earlier case, because this is involving specific chemical interactions with biological molecules, it can be called as bioaffinity. Gel filtration is the technique where we use a column to separate molecules on the basis of the shape of the molecule and the molecular mass. Whereas ion exchange depends on the surface properties of the protein at a particular pH. So, depending on the pH of the protein and the pH of the buffer, the protein can either adopt a fully positive charge or a net negative charge. So, we use cation exchange and anion exchange. Right. The next question is, Ni2 plus is used to purify proteins with dash. For this question, what one can do is, although we have discussed the right answer several times, you can try thinking what the other tags could play a role. There are streptavidin tags, NUS tag, and MIC tag. While we do experiments, we think about adding tags to a protein to serve different purposes. It could be to solubilize proteins, it could be to secrete proteins. If anyone has any idea as to which of these uh, tags are used for which of these purposes, you can share that with us, because it's interesting that different properties of the tags help in different functionalities within a cell. You can take a couple of minutes to think about the different incorrect options here.
Sir, A.
Yeah, A is the right option. We know that his tag is used for nickel affinity. What we can you can take a couple of minutes to think about the wrong options. We'll also learn what the incorrect options are and why a particular tag can be used for what purpose. So if you know what streptavidin tag is, or NUS tag, or MIC tag is, you can take your time to think about it and then answer. Okay, we learned that a 6x cysteine tag can be used for purifying proteins with nickel and beads. Streptavidin is something that can be selectively bound to biotin. So, if there is this process called as biotinylation, where whatever molecule of interest can be uh, attached covalently with biotin. So, what people do is they either add a streptavidin tag and then have a bead with biotin and then elute it. NUS tag represents a protein called NUS, N utilization site, which you might have heard during your learning period for protein transcription. Now, this protein helps in solubilizing any protein. So, let's say you are trying to purify a protein and the protein is precipitating heavily. This could mean that the protein is not folding very well. When you do add tags like GST tag or NUS tag, it helps in improving the solubility of the protein so that you get larger yields of the protein, especially for biophysical experiments or structural characterization where you need lots of protein. And finally, MIC tag. MIC tag is something that is used for secretion of protein to the exteriors of the cell. This is just a representation for nickel NDA. Here is what I meant as the three-dimensional coordination geometry. The next question is, in irreversible affinity chromatography, the ligand receptor pair interacts with dash. And four options are given here: covalent bonding, hydrogen bonding, van der Waals interaction, hydrophobic interaction. So it's irreversible, not reversible. So you can think which of these four options is irreversible. And once you know that, you can mention it.
Sir, covalent bonding because it's irreversible.
Correct. Yeah, so there are two types of interactions that we could see with respect to bonding. One is covalent, one is non covalent. In covalent, what happens is you have a physical bond that is being formed and that can be broken down either by so you need a lot of energy to break it, and usually what people do to break this is use enzymes. These cannot be denatured efficiently by heat or application of other chemicals that can denature your non-covalent bonds. Whereas your non-covalent bonds are one of these types, either electrostatic or van der Waals or pi-pi interactions or hydrophobic or hydrophilic interactions. Here we see that there is electrons moving around that are delocalized in pi-pi interactions and form these parallel stacking interactions, or this could be due to partial charges like in the case of van der Waals, or it could be movement of partial movement of charges, like in the case of hydrogen bond due to electronegativity. In all these cases, the bond that is being formed is not physical, rather, that can be disrupted. But in covalent bonds, it is physical. A peptide bond is an example of one covalent bond. Right. The next question that we look at is lysine is used as a receptor to purify dash, and four options are given here: poly U mRNA, rRNA, glycoprotein, immunoglobulin. So, again, we are trying to look at affinity, specific affinity. So, I'll just clarify a few things so that you can think what can have good affinity. So, poly U mRNA means it has a stretch of multiple U's, and we know the logic of base pairing. So, another stretch of oligonucleotide with a particular base can help in improving or binding to this mRNA. rRNA, unlike mRNA, unlike poly mRNA, is non-specific. It can have any bases. So we should not be dependent on bases. Rather, we should look at some other properties that are abundantly present in the RNA. One clue that I can give you now is the RNA has this phosphate backbone which is negatively charged. The third one is glycoprotein. Now, glycoproteins are proteins with your carbohydrates attached at particular sites, and immunoglobulins are your antibodies. So you can think in these aspects and try to justify why lysine, which is a positively charged amino acid, which can be used as receptor for one of these or more of these. You can think about this question for a couple of minutes and then we'll discuss this. Does anyone want to try answering? Okay, I'll go through the right answer. So poly-U mRNA, like I told, it has multiple U, which is uracils, at one of the ends, usually at the 3' end. So because this has multiple U's, if we have oligo DTs, which is multiple T's, and because of base pairing, we can we'll be able to purify this. Glycoprotein, these are purified by lectins. Lectins are nothing but plant proteins that can bind to your sugar molecules. Since glycoproteins have these sugar molecules attached to this, that is one way of selecting this. And immunoglobulins are purified by proteins called as protein A or protein G. Now, RNA, like I mentioned, because rRNA doesn't have any of these specific properties like poly-U, we need to look at something that is globally present, which is the phosphate backbone, and that is negatively charged. So, using lysine cephalose or something like that, we can use this positively charged lysine with negatively charged RNA to be able to bind it. So, that is how lysine is used as a receptor to purify poly-U mRNA. The next question is, carboniimides helps to immobilize DAS functional groups in receptors: amine, carboxyl, hydroxyl, and esters. You can take a couple of minutes to think about this question, and then we'll look at the right answer. So, what we need to understand is these carbodiamides, they help to add a particular functional group. Now, these are organic reagents. What it does is it forms these, uh, it helps in creating covalent bonds. So, where these are used is in experiments like SPR, surface plasmon resonance, where we have some chips and we want our proteins to be bound to that chips. So, what we do is we treat it with these chemicals. So, what happens is this carbodiamide it reacts with the carboxyl group to form this unstable acyl isourea intermediate. Then the activated carboxyl group reacts with the amide to form an amide bond. This way, the protein is embedded on the chip, which can later be used for other assays. So the right answer is it helps to immobilize this. It's an amide, not amine, functional group present in receptors. The next one is GST tags binds to dash immobilized on the matrix. Glutathione, glucose, glycogen, glutaraldehydes. Okay, for this, you can try thinking what GST could mean. That is a protein that is used as a tag. Unlike your his tag, this is not small. This is rather a complete enzyme. And we use this enzyme's functionality because this enzyme is can fold well and can solubilize well. We use this with at the N terminal or C terminal of a protein of interest. So, does anyone want to try what GST could mean? GST is glutathione transferase. That is an enzyme. So, like I mentioned, because this is a very stable enzyme, you put it either at your N terminus or C terminus, and proteins that would not solubilize to higher concentrations. When I say solubilize, what I mean is as you concentrate your protein to higher concentrations like 10 mg per ml, 20, 30 mg per ml, you would start seeing small precipitates. This is because of aggregation of proteins. In some cases, this happens even at 1 mg per ml or 2 mg per ml. So people use tags like GST and NUS. And apart from helping in solubilize your protein, GST can also be used specifically to elute. So that is shown here. So this is our protein with GST. And we have this glutathione affinity matrix. So when we add this reduced GSH, which is reduced glutathione, that would compete. And this competing GSH would bind to the GST tag. So that would release our protein from the glutathione affinity matrix. So this way, our protein of interest will be eluted. The next question is, booster injections result in the formation of dash. Option A, monoclonal antibodies. Option B, T cells. Option C, macrophages. Option D, memory B cells. So to start with, for this question, you can tell me what booster injections are and when are these injections given. Then we look at the different options that are given here. booster injections, we'll first discuss what booster injections are. So these are injections given other than your prime injections. So in your cases, when you were uh, exposed to vaccines during your vaccination schedule, you would have been given more than one injection for the same disease at a different time point in your life. So, what is expected is in your first injection, which is your prime dose, you are exposing your body to the antigens. The second dose is called the booster. Here, we re expose the immune system to the antigen. This is done so that the immune response is in such a way that it lasts for many years. Now, the question is what does booster injections result in the formation of? And four options are given here: monoclonal antibodies, T cells, macrophages, and memory B cells. We'll eliminate these options one by one. Now, first, monoclonal antibodies. Now, these are specifically created. Monoclonal as in all of these are derived from one particular, it is for one particular antigen produced from one particular cell. So, this is similar to how you say uh, specificity, just one particular antigen and one B cell produced an antibody against it, and this is used as a clone and this is propagated. But this can happen only within the, outside the cell, in vitro. So, monoclonal antibodies should be removed. T cells and macrophages, these are again naturally present within your body. T cells you have as helper cells and so on. Macrophages are freely roaming and they come to site of infection like your neutrophils. So these are not result of booster injections, but booster injections help your B cells, your memory B cells. So they convert your B cells to memory B cells and ensure that at any point later in your life, if you have any infection with the same antigen, these antibodies should be, some antibodies should be produced. So that is the purpose of this booster dose. So to sum it up, the main outcome is the production of long-lived memory B cells, which allow for a much faster and stronger antibody response upon re-exposure. The next question is, again, this is similar to what we looked at earlier. Affinity chromatography can be used to identify the interacting partner of the dash. Option A, analyte. Option B, buffer. Option C, urea. Option D, matrix. So here your buffer has nothing to do. Your buffer will only ensure that your molecule of interest is uh, not altered by, there is no alteration of pH. Urea, we use urea for denaturing, and again, that doesn't have anything to do with affinity chromatography. Matrix is your solid support. Like for example, in nickel NTA beads, your agarose is your solid support, onto which you have nickel in nickel. So the right answer here is analyte. Again, one more simpler question. Dash is used for blocking of non-specific sites on the beads of a column. Salt, receptor, ligand, and BSA. So when it comes to non-specificity, is there something that comes to your mind? Has anyone here done something like a Western blot or something, where we try to put a gel and then transfer the contents of the gel onto a membrane, and to prevent non-specific interactions, we cover it with something called as a blocking agent? Does anyone here know about these things?
We use probes for specific binding.
Sorry, what do you use?
Probes
What is a probe?
in vegetable where we need a specific want to bind to specific protein.
Yeah, so now let's say we want to block non-specific sites. What do you think can be used? A probe could be anything. It could be something that contains a fluorescent tag, or something that is used in amplification, like a primer. A probe could be anything, but that will be specific. Now the question is about blocking non-specificity. The reason I talked about Western blot is, in Western blot we usually do have this step called blocking step, where we ensure our uh, that whatever we want to identify should only be exposed, and unwanted molecules should not be amplified in the background.
DSA.
Yes. So there we use skimmed milk powder.
Okay.
We add those proteins. So we expect that, um, again, your milk will of course have um, all the other proteins. Here we would use BSA, because bovine serum albumin, that is one protein that is used for multiple purposes, like to improve stability and so on. In this case, we use BSA because that would have these charges that could compensate for non-specific interactions. So when we put our protein of interest, only our protein of interest could bind to it, and other random proteins would most probably not bind. So that is why here the right answer is BSA, for blocking non-specific sites on the beads of a column. The next one is an interesting one. To identify the peptide sequence of a protein after affinity purification, dash is used. Mass spectrometry, UV spectroscopy, fluorescence spectroscopy, and circular dichroism. Now, for this question, independent of whether you know the right answer or not, I would expect a few of you to unmute and tell me what you know about each of these techniques so that we learn where exactly each of these techniques can be used. You need not know the right answer specifically for this question, but if you know any of the options here, you can tell me. We'll discuss that so that all of us can learn. You can take a couple of minutes to think about this and then we'll discuss.
So spectrometry is used for the chemical structure
Yeah,
and
so, yeah, you can continue. Sorry. So you're saying mass spectroscopy is used for? It's actually mass spectrometry. There's no spectroscopy
okay.
here. Yeah.
A spectrometry is used for the chemical structure.
No, no. What do you mean by chemical structure?
In chemistry, sir, we determine the from the um, we can draw the structure with the help of mass spectrometry of any chemical compound.
What do you get in mass spectrometry? What do you measure in mass spectrometry?
Mass
Yes.
uh,
So with the
molecular
mass, will you be able to tell the chemical structure, or will you be able to tell an approximate chemical composition?
approximate chemical composition.
Right. So you'll not be able to get the structure.
Mm.
Right. Do you also know about any of the other options? UV spectroscopy, fluorescence spectroscopy, circular dichroism.
The
UV spec-
cycler dichroism is used for conformation changes.
Okay. When you say conformation changes, can you be a bit more elaborate?
Structure
Conformation.
like protein beta helix, alpha.
Okay. So, do you know what happens in circular dichroism? Like, what component do we use? What is the kind of light we use? Plain polarized, or if there is specific absorption and the reason for specific absorption?
No, sir.
Anything to do with chiral nature of amino acids, D, L, and any idea on that part?
No idea, sir, about. I know about the conformation change,
Yeah,
like
yeah, it is
that's it.
of course related to conformation. So, what we know is the light that we normally use is not polarized, but plain polarized light will have the different components only at two different angles, not in all 360 degrees. So, because of
I remember
this,
like
yeah,
circularly polar, like right circularly polarized light and left circular polarized light, sir. I remember that part somewhere in
yeah,
seed.
good, good, true. So, we have this polarizer that splits your light, and depending on the kind of chemical structure that is present, whether it's an alpha helix or a beta sheet or an unstructured loop, this will be absorbed differently, because of which you will have peaks at certain wavelengths like 192 nanometers, 220 nanometers, 208 nanometers, and so on, specific to specific secondary structures within a protein. So when we do circular dichroism, and if we have protein that can change its conformation over time, what we can, or in the presence of a ligand or so, what we can do is we can measure the absorbance at specific lambda values appropriate for the alpha helices and beta sheets and so on, and measure it in different conditions to see if those conditions has altered the folding of the protein. And that is what you meant by conformation. Good. Whereas UV spectroscopy is used to know. So now majorly people use it for uh, calculating the concentration of protein. But in addition to calculating the concentration of proteins or nucleic acids, UV spectroscopy can also be used with small molecules to get again their concentrations. Fluorescence spectroscopy is a different method where we excite our protein molecule of our molecule of interest at a particular wavelength and um, see for emission. Now again, people use this for quantitative purposes, but none of these will help us tell the peptide sequence. So the only method that we can use is mass spectrometry. So here in mass spectrometry itself, there are two methods. One is TOF and one is MALDI. So in TOF, we have this method called time of flight, where we just allow our protein to fly, and depending on the energy, the voltage that we have, and the velocity and the time that it takes to reach the other end of the detector, we'll be able to calculate the mass. So with this we can get an approximate size of our protein, but this doesn't tell the sequence. To be able to tell the sequence, what we do is we digest our protein randomly with a protease, and we will have different fragments of our protein. We ionize it and we allow them to fly. And once we allow them to fly, in the second step called tandem mass, that means there is two steps of this mass spectrometry that is happening. What happens is we get peaks for different masses, different m by z ratios. Now these m by z ratios can be correlated with different lengths of amino acids with sequences with 2 amino acids, 3 amino acids, 4 amino acids, and so on. So with an existing database that contains all combinations of amino acids for all different lengths, we try to correlate and get the length of try to get the approximate sequence of the protein. So this is uh, one method that can be used to identify the sequence of a protein after affinity purification. Was that clear to everyone?
Yes,
Yes,
sir.
sir.
Okay, good. Okay. The next question is, cytokine levels of a lymphocyte can be measured by DASH after performing affinity chromatography. And we have four options: MALDI, TOF, ELISA, and hybridoma technique. Now, before we go on to the right answer, if anyone knows the full forms of these different methods, you can mention it. Or if you know any options here, you can still talk about that option. We can discuss that later whether it is right or wrong.
sir, I know about ELISA, sir.
Sure, yeah, you can tell us what you know about
The
ELISA.
full form is enzyme linked immunosorbent assay,
Right.
and it is used to detect the antigens or antibodies in a sample.
Okay. So, do you know how this is performed?
Yes, there are three types of ELISA, like direct ELISA, indirect, and sandwich ELISA. There are three types.
Okay.
So first antigen is immobilized on the plate,
Hmm.
and then enzyme linked antibody binds to the antigen.
Okay.
So we can knew it like that. And
Now,
we also horse. I don't know enzyme name. I just
horseradish
forgot. Yes.
peroxidase. Yes.
Yes, sir.
Yes. Yeah, that comes under secondary antibody and detection. So, what exactly do you want to detect there? You told antigen or antibody is coated on this plate, it is immobilized. So, then you say that you have this antibody that binds. So, but what exactly do you want to detect there if that's the case? Is
We
it
want to detect
is it an
antibody.
interacting partner that will bind with your immobilized material? Is that what you want to detect?
Mm,
Yes, that's the right answer. You can tell it confidently, no issues. So we
yeah.
have so ELISA is a quantitative method. People use that to quantify whether binding is happening. Um, Based on the value of luminescence, you can quantify in ELISA. So, like you had mentioned, we have a plate which is immobilized with an antigen of interest. Now, let's say two different people are infected with the same pathogen. They will have different quantities of antibodies depending on the nature of the person. Now, if you take the blood sample and take the serum and get the antibodies out, and if you pour equal volumes, both of them will have different readouts in the ELISA, meaning one will have higher, one will have lower. Now, ELISA can be used to quantify and tell us how much is present for each person. That is where ELISA is used. Good that you shared what you know about ELISA. Do you also happen to know about MALDI or TOF? We just discussed about it earlier, or hybridoma technique.
Hybridoma technique. I know a little bit, sir.
Sure, sure.
I know it is used to produce the monoclonal antibodies.
Okay, so what do we do there?
As a processor, it involves in the fusion of antibody producing B cells with myeloma cells to form a hybridoma cell line.
Okay, so we take a B cell that will
B cell, and we
produce
are
only
going to fuse
one
it to myeloma.
antibody or for one particular antigen, and then we fuse it with a carcinoma cell so that it can infinitely produce the same antigen, right?
Yes, sir.
Good, good. Yeah, like he had mentioned, these are the two different techniques, ELISA and hybridoma. MALDI is what we TOF is where we allow particles to fly for a given potential difference or voltage, and then calculate the mass from the time it takes to flight, TOF, time of flight. Whereas MALDI is the method where it is abbreviated as, it is expanded as matrix assisted laser desorption ionization. Here we take our macromolecule of interest and we embed it on laser, embed it on a matrix and hit it with a laser. It's also a part of your mass spectrometry. When we hit it with a laser, it ionizes. Now, coming back to the question, the cytokine levels of a lymphocyte can be measured by DASH. Like I mentioned earlier, when we have two people with having the same infection, we can measure the antibody response in both the people using a method called ELISA. Now, in case we are not looking for antibodies, but for cytokines, that can be modified by just changing the substance that is immobilized. So, ELISA is the right answer for this question. Okay. The next question is, antigens are isolated in denaturing conditions using dash. Electroevolution of antigen from STS page, gel filtration chromatography, ion exchange chromatography, affinity chromatography. You can take a couple of minutes. Now, an additional information that I can give you is, let us say I have an antigen antibody complex. Now, I want to remove my antigen alone. What should I do to remove my antigen alone? All that I know is my antigen antibody complex is very large, my antibody is very large, my antigen is relatively small compared to my antibody. So, which of these techniques do you and they are bound, they are physically interacting. So, which of these techniques do you think will be helpful in isolating them? You can take a couple of minutes to think about this and then we'll discuss. like I mentioned, now we have an antigen antibody complex which is tightly bound. So, what is one way in which we can first prevent this interaction? Does anyone want to try answering? Do you think gel filtration or ion exchange or affinity chromatography will prevent this interaction? Okay. The only way in which I think we can prevent this interaction is by running it on a denaturing page. So molecules of different molecular weight will get separated. And once they get separated, we definitely know what an antibody would weigh, based on which we know what our antigen would be. So if we electroelute, we'll by electroelute what I mean is we cut the STS page, the gel, into pieces and try to extract the protein from it. So if we electroelute and then mass spec and get the sequence, we'll be able to isolate the antigen. Why can't gel filtration chromatography work? Now the thing is, this is already a tightly bound complex. Unless we denature it, we'll not be able to separate the antigen from the antibody. Why can't ion exchange chromatography work? Again, ion exchange chromatography works depending on the pI of the protein and the pH of the solution. Because we have no idea about the antigen, we don't know if it shares the same pI as our antibody. So, setting the pH and putting cation or anion exchange chromatography without having an idea about the pI of the protein, which is the antigen, is of no use. And finally, affinity chromatography. We don't know if, even if we allow our protein A or protein G beads to be able to bind to our antibodies, we don't know if antibody will still be bound to our antigen or not. So, because these three methods will still not prevent the physical interaction between your antigen and antibody, the only way that I think this can possibly be done is by running it on a denaturing page. It could be an STS page or a urea page, but it would work. The next question is, proteins with DNA binding sites can be purified with the help of dash heparin coupled matrix, CNBR activated polysaccharide matrix, biotinylated matrix, streptavidin coupled matrix. You can take a couple of minutes to think about this. Now, one thing that you need to keep in your mind is the charge of DNA. DNA is negatively charged, and proteins with DNA binding sites will be positively charged. So this means that your affinity will be towards something that is mimicking your DNA. So you need to think which of these will be negatively charged, and then we'll be able to get the answer. earlier we had discussed about biotinylation and uh, streptavidin. So these complement each other. Whereas heparin is what people use for proteins with DNA binding sites, because heparin is positively charged and DNA is heparin is negatively charged, and it mimics your DNA phosphate backbone, and proteins with this can DNA binding site will have a positively charged, so it can bind to heparin. And the last option is CNBR activated polysaccharide matrix. These are used for covalent coupling of proteins or ligands and not for DNA binding proteins. The last question for today is non-specific binding of immobilized receptors with the analyte protein is done by dash. Equilibration with NaCl, imidazole, urea, or guanidinium hydrochloride. You can think about this question for a couple of minutes and then we'll discuss. So for this, you need to think about the role of each of these solutions. What can NaCl do? What can imidazole do? What can urea do? What can guanidinium hydrochloride do? So NaCl will change your properties of protein-protein interaction. At different concentrations of NaCl, you would see the same protein having different oligomeric status. So when that is possible, it means that protein-protein interactions will be inhibited. Imidazole is a competitive binding analyte for histag proteins. Urea and guanidine hydrochloride both can denature your proteins. So here, non-specific binding of immobilized receptors with the analyte protein is done by equilibration with NaCl. So with this, we come to an end for today's class. If there are any questions, you can ask me. I'll stay on the call.